Good afternoon. It's now my pleasure to introduce our next participants from Senti Bio. We have Dr. Timothy Liu, co-founder and chief executive officer, and Dr. Philip Lee, co-founder and chief technology officer. So welcome. And the format for the session is approximately 25-minute fireside chat, but first we'll start off with a few high-level overview slides of Senti and its manufacturing capabilities, and then we'll move on to Q&A. As a reminder, if you have questions during the session, feel free to type them into the chat box in the Zoom window, and we'll try to ask them on your behalf. So, Tim, Philip, please begin.
Great. Well, thank you to Chardan. Thanks, Gula, for the invitation, and we're really excited to be here today to share with you Senti's gene circuit platform, as well as our efforts to translate these into products for patients, along with the manufacturing that's going along with that. Senti Bio is a biotechnology company based in the Bay Area in California, and it's focused on applying synthetic biology to tackle key challenges in the cell and gene therapy space. Really core to what we do at Senti is developing what we call genetic circuits, and these gene circuits serve as the intelligence inside of a wide range of cell and gene therapy products. At Senti, we've built gene circuits for many years, and we have developed a technology platform that allows us to create cell and gene therapies with potentially enhanced efficacy, safety, and control through the design of these gene circuit elements. We've shown that these gene circuits can be inserted into a variety of different therapeutic modalities. Internally, Senti is focused on NK cells for oncology, with anticipated IND filings in 2023. Focused on Senti 202 for AML, as well as Senti 301 for liver cancer. But these gene circuits, similar to genome editing, can be put into a wide range of applications. And so we have built gene circuits that function in, for example, T cells, gene therapy vehicles, et cetera. And we've adopted a strategy of partnership with leading collaborators in areas outside of oncology. These include partners such as Spark, as well as with Blue Rock. Because today is focused primarily on the biomanufacturing side, I won't go into a ton of detail into the gene circuits that we've built. But at a high level, there are four major categories of these gene circuits that we believe tackle major unmet needs in the clinic. Logic gating, for example, we can design cells that actually recognize multiple antigens rather than just a single one. And based off of which combination of those antigens are present will then trigger killing or protection of the target cells. So these include gene circuits like an OR gate, where you can build a CAR-NK cell that recognizes any one of two different targets. Either one will trigger killing, thus increasing the spectrum of treatment. We've also built what we call the NOT gate. The NOT gate allows us to protect healthy cells from being killed, essentially an artificial don't eat me signal. So logic gating is really centered around this idea of enabling more specific and targeted therapies while protecting healthy tissues. We have a second class of gene circuits called multi-arming, where we can design cells that actually not only contain a CAR, but are also going after multiple other mechanisms in the tumor microenvironment. These include controlled release of cytokines, as well as combinations of cytokines that are being produced to amplify the anti-tumor immune response. We have a third type of gene circuit called the regulator dial, which is a gene circuit that allows us to control what a cell and gene therapy is doing, even after it's been delivered into the body. And that's done using a small molecule drug that can be consumed orally. We've optimized these systems to make them highly controllable and sensitive to the small molecule FDA-approved drug, and that allows us to regulate the activity of these products. Finally, we've built different classes of smart sensors, including promoters that can detect when and where these cell and gene therapies should be active. As I mentioned earlier, we've been applying this into a wide range of modalities. Internally, we are very much focused on off-the-shelf allogeneic CAR-NK cells with this sort of approach. So specifically, we are applying these logic gate and multi-arming gene circuits to go after disease areas where there is not a single clean target to go after. This includes diseases like AML, colon cancer, as well as other solid and liquid tumors. So by implementing our OR gates and our NOT gates, we can more effectively separate healthy cells from cancer cells and therefore pinpoint all of our activity on those healthy cell populations. We're also introducing our multi-arming gene circuits and our regulator dials into products such as Senti 301 for liver cancer, to not only target liver cancer cells for killing, but to secrete cytokines that amplify the activity of those CAR NK cells. Below the middle line here, as I mentioned earlier, we have collaborations with great partners. In these situations, Senti is primarily designing the genetic circuits and working with our partners that have manufacturing and clinical development teams to take these forward in downstream indications. We like CAR NK cells as a modality for our gene circuits for a variety of reasons. Natural killer cells have innate killing activity as well as the natural ability to activate the immune system. And so we can leverage both of these features to really improve their efficacy in terms of doing that in patients. I think a lot of clinical data has now been accumulated suggesting that these can be used as an off-the-shelf, safe therapeutic for oncology, in many cases with a much better safety profile than T-cell-based therapies. And Philip will talk later about a lot of the work that we've been doing to ensure that we can manufacture these gene circuits with our genetic circuits in them to enable potentially broad access for patients. There have been now many clinical studies, both academic as well as industry, including some news that came out today, showing that NK cells can have good functional activity in humans. So our process for modifying our CAR NK cells with our genetic circuits looks as follows. We currently isolate NK cells from healthy donors. We use viral vector to basically transduce our gene circuits into the cells. Now, one of the things to note about our gene circuits is that they typically involve multiple genes. These are larger constructs that allow us to implement all of the sophisticated sense and respond activities that we're putting into these cells. So the gene modification step is critical, and we've developed a lot of know-how in terms of how to do this efficiently. We can then expand the cells, freeze them, and distribute them with the idea that these cells retain their functionality even post-thaw. Our strategy is to take our CAR NK pipeline programs into the clinic and demonstrate key proof of concept data in patients. Senti 202 and Senti 301 are our two leading candidates. And as part of that, we've decided to manufacture and develop these NK cells in-house. Much of the know-how that's important in growing NK cells, expanding them, and importantly, transducing them with our gene circuits, as well as building the analytical strategies for assessing gene circuit functionality are very important for us to keep internal to Senti. It's central to the quality metrics that we're building into these products. We have our labs, our preclinical labs in South San Francisco, as well as access to a GMP facility in Alameda that is being built out to support this in-house GMP manufacturing for downstream applications. So with that, we're really happy to be here today. I wanted to give you an overview into the gene circuit technologies that Senti has been building. Specifically for NK, as I mentioned, we are continuing to manufacture and develop these cells internally for clinical studies, in addition to the partnerships we have in areas outside of oncology. And so with that, thanks for the time and happy to turn over to Gula.
Great. So that was a really nice overview. And maybe to start off, we can talk about areas that you see your approach in terms of manufacturing being differentiated from other companies in the space that are, as you mentioned, also advancing cell therapies and in particular NK cells.
Right. Yeah, no, happy to start off with that answer. I think, as Tim mentioned, one of the key things for Senti as we thought out of building out our manufacturing strategy is how do we encompass the capabilities of the various gene circuits into different modalities, and particularly into the NK cells for us. So probably, just from a strategic perspective, we are very much focused on having close integration between our gene circuit design teams with our process development analytical teams, all the way into manufacturing, as Tim mentioned, with our in-house manufacturing facility. And what turns out to be true is, a lot of the circuit capabilities, these multi-target CARs or the regulated controllers, require sophisticated analysis to figure out, how to tune them properly and how to design the best capabilities. So our analytical capabilities, starting from an early stage is very important, all the way through how we scale up the process and then, into the manufacturing piece. I'll pass it over to Tim to talk a little bit about some of the specific gene circuits that are relevant to the CAR NK space. But again, I think, the ability for us to take those capabilities and integrate them early on into the development process is something that's very important for us.
Yeah, I think, as I mentioned earlier, really the key secret sauce for us is how the genetic circuits function. The logic gates that allow us to sense multiple antigen targets, have the cells make a decision. All of those require us to build vectors that have multiple genes that are in them. So we certainly have a lot of know-how in terms of how do we design and get these to function, but then we have to go through the practical issues of how do we get these into cells, how do we manufacture them, et cetera. So we view these as intimately linked. We oftentimes have changed the design of the genetic circuits to try to enhance the manufacturability of the downstream product. You may not think about it that much, but depending on whether you put one gene ahead of the other, or put certain other sequences in between, can change the effectiveness of how well it's expressed, how well it transduces, how well it functions. Even though the proteins should be the same, but the actual vector design makes a big difference in terms of the downstream process and manufacturing steps.
Yes, that makes sense from my days in the lab. Yeah, no, so actually, so on the topic of the gene circuits and integrating them within your manufacturing, on the flip side, can you use synthetic biology to improve manufacturing in other ways?
Yeah, so I'm happy to start there, and Philip, you should weigh in. So there are really interesting applications, I think, for synthetic biology in the broader biomanufacturing space. And we have demonstrated, in publications, for example, that we can build gene circuits that allow us to control glycosylation in CHO cells for antibodies, for example, and be able to tune up or tune down, how much of a particular pattern that you're seeing. We think that there are similar use cases in, for example, AAV biomanufacturing, where one could potentially engineer those cell lines to give you control over the biomanufacturing process, as well as control over AAV gene expression. And certainly in the cell therapy setting, beyond the sort of vector design itself, oftentimes many of these cell therapies are using, for example, feeder cells as part of the process, and there is genetic engineering that goes on there. So we think it is an area of tremendous opportunity and interest, and I think we believe that the field overall is starting to see this. So I do believe that that's an area that's going to grow in the future as well.
Yeah, I think a couple examples, with our current NK programs where we see the synergy between the gene circuits and the manufacturing. One is in terms of cytokine engineering, in terms of expressing these from the NK cells. I think one of the things we found, if you think about the NK cells, is that the IL-15 support is important in terms of activation as well as expansion of the cells. So our ability to generate a engineered version, which we call the calibrated release IL-15, I think has been both impactful in terms of getting the cells to function better. But when you look at the actual manufacturing process that enables, that does give you ways to tune your process development, your scalability, and ultimately how you characterize the products. That's helpful. A second example, in the 301 program, where we're controlling the expression of IL-12. So as Tim mentioned earlier, being able to dial that in terms of the off-target expression when the off state, how much expression you have, and then the on state, how much expression you have. Those are things that are also important to, as you think about the cells expanding, during the manufacturing process and ultimately, being delivered as a therapeutic, having a good handle on how these cytokines are expressed, when they're expressed, and control over, kind of the on levels and the off levels is, something we found to be very useful in terms of a robust manufacturing process as well.
So you mentioned earlier that the gene circuits are portable to other cell therapy modalities and vector modalities, et cetera. How does that play into manufacturing? And does your facility have the flexibility to also manufacture for other modalities?
Yeah, I can talk about the facility piece. It's a key point, and Tim can comment on some of the other programs we're working on outside of NKs. So one of the things, as we, even when we started thinking about our CMC development teams, we wanted to have broad expertise across different cell and gene therapy modalities. Understanding the direction of where the technology is taking us. I think as many people here understand, even if you think 2 years, 3 years ago, the types of modalities, the types of cell and gene therapies out there have evolved quite a bit. So we do want to be prepared to quickly adapt as the technology and as the field adapts. So we have a CMC team that's very knowledgeable across multiple modalities, multiple cell types, have worked in different therapeutic or clinical products as well. And when we thought about how best to design our manufacturing facility, we did want to adopt that approach too, in terms of modular clean room setup, where we can have segregation of different rooms as well as different wings of those rooms. If we want to have multiple products of different modalities, we can handle vector manufacturing, viral vector manufacturing, as well as primary cell therapies. So that gives us a lot of flexibility in the sense of thinking about both future scale up in terms of capacity, as well as different products and different modalities that we can hopefully quickly adapt to, as our portfolio expands.
Yeah, the only thing I would add to what Philip said, and he made a really good cogent argument for a modular facility and team on the manufacturing side, but we've also been doing that on the upstream side, on the research side as well. So the way we design these gene circuits is usually not a one and done sort of thing. What we want to do is build these modules so that, yes, we're designing them for NK cells right now, but much of the learnings from that process could be carried over into other cell types. So for example, the regulator dials that we've been building, that basically allows us to sense a small molecule drug and change gene expression. That's a very general thing that would be useful for, cell line manufacturing, like you mentioned earlier. It could be useful for CAR Ts. It could be useful for gene therapies or iPSCs. And so when we do the experiments for designing these gene circuits, we spend a lot of time making sure we have, rigorous capture of the data that's being generated, so that the next time around, if we have to go back to that data set or redesign or improve the efficiency of the system, we have already a good base to learn upon. So I think both the early stage as well as the downstream manufacturing, we've tried to build in as much sort of engineering and learning as through the process as we can.
Got it. That makes sense. And so you mentioned earlier that the gene circuit constructs and the products often contain multiple components. So I'd like to understand a little bit more about what that means for evaluation of things like potency, composition, and consistency. That's things that regulators seem to be paying attention to in this space.
Yeah, I think, what we see, and, I think, everyone we talk to at conferences nowadays see is, the field is definitely moving in this direction of having multifunctional therapies. I think a lot of the reason some of these cell therapies have such great effects in the clinic is because inherently cells do have multiple functions. Even with a non-modified NK cell, as an example, they are doing a lot of, sensing of their environments, acting on different cell types, as well as expressing different factors. So I think there is inherently this combination of factors that's at play with these therapies. I think for us, you mentioned the regulatory piece. It essentially comes down to, sort of a data-driven approach. How can you demonstrate consistently that your product is performing, from an engineering spec perspective, the way you designed it to? So for us, we spend a lot of time thinking about the analytical characterization, going back to when we design the circuits, how do we even detect that they're working, and, what are the sort of handles that we want to start to measure? And then at the end of the day, I think, at least our strategy and what we've heard in the field is, if you can show consistency in terms of the manufacturing and the CMC perspective, you have ranges that you're comfortable setting for your product, even if it is multiple components, that's something the FDA and other regulators are familiar with. But it does require, sort of an additional level of analysis and making sure you're looking at all the different components, both independently as well as in combination. And that's something, we've spent a lot of time over the past couple of years really characterizing in terms of our gene circuits. The other thing, just to quickly add, is the benefit of having engineered functions into the cell therapies is that, as the designers of those functions, we understand what they should be doing. So we know, what genetic sequences to look for. We know, what effects or what potential, effects you don't want to see. So I think, from an engineering perspective, that's something we see as continuing to be the case where, as these cells become better understood and we're better able to program in the functions, that actually makes it easier for us to characterize the cells and maintain consistency and safety for the clinic.
And in terms of scale, where are you at in terms of the number of doses that can be produced, or whatever other metric that you're currently using in terms of scalability, and how do you envision that the need for that potentially evolving as you advance towards commercial scale?
Yeah, great question. I think the field itself is moving very rapidly, which is very exciting. I think as Tim alluded to, every couple months, I think we learn something new. For us, our strategy is kind of in parallel, getting the key gene circuits into the clinic to demonstrate or to understand how they function, and then in parallel also developing the next step in terms of scalability and commercial manufacturing. So, with an allogeneic off-the-shelf NK cell, I think as many folks have seen in the clinical space, right now, we believe targeting something in the hundreds of doses per batch is very viable in terms of, demonstrating what the actual manufacturing process should look like, how to scale that, as well as, ultimately getting that clinical result to inform dosing, to inform, kind of how the process behaves. So I think for us, that's very much our core focus in terms of the initial clinical programs. We have also been working on thinking about that commercial phase of what technologies are available and what can we further develop to get to, call it a couple of orders of magnitude larger scale. And I think as, some other presentations have shown, just in NK biology itself, there is a lot of room to play with in terms of how you get NK cells to expand, how you can adapt them into larger bioreactor type systems. And we're pretty excited about that looking forward.
Great. And so I guess it may be tying to some extent in the topic of scaling. What do you see as the major cost drivers for CAR NK cells? And are there any particular innovations that you're looking at that can reduce or circumvent some of these major cost drivers?
Yeah, the allogeneic cell therapy field is pretty new. There's clearly no approved products yet or no commercial products. So I think we're all looking at a lot of these technology developments eagerly and helping develop them. I'd say at the end of the day, from a math perspective, essentially, the number of doses per batch is what's going to drive cost. So that includes how much can you get the cells to expand, how quickly do they expand, as well as your, recovery rate, both through the harvest as well as in this crop preservation process. So I think, what I've seen is there's been a lot of improvement over the last years in terms of instrumentation, process equipment, and just general know-how of how, to do that more efficiently. And we anticipate, over the next couple of years, as more allogeneic products hit the clinic and, we all understand how to process them better, this will continue to move in that direction of reduced costs of manufacturing, as well as, larger batches and consistency. In terms of specific technologies, we think the bioreactor technologies have come a long ways in terms of adapting to cell therapies, which is different than sort of biologics manufacturing. And then also the analytical equipment is very important, specifically, cell analysis and single cell analysis. I think it's going to become increasingly important.
Fantastic. So in the last couple of minutes, I wanted to get your perspective on what you think people might be underappreciating regarding Senti's CMC capabilities.
Yeah, I think for me on the manufacturing side, I think what we mentioned earlier of how we thoughtfully integrated from design all the way through into the GMP and even into the commercial phase, having that sort of integrated approach has been very important for us. And we think that'll continue to be a key aspect of our development moving forward for Senti as a company. Maybe Tim, if you want to add anything to that.
Yeah, that's a good question. I think I've been working in the synthetic biology field for close to 2 decades now. Most of the work that's been demonstrated, we've seen really exciting results in the lab. But I think the gap between taking something that works well, in a small scale and being able to scale to large scale, there's a huge gap there. So I think one of the things that Philip and his team, along with our internal research team, have really integrated quite well is to, like I said, we're not just designing gene circuits that work at the small scale, but when we learn which ones are working well at the process development and scale up levels, we can go back and redesign and try to optimize. And so I think from a competitive perspective, that is a lot of know-how that's very hard to teach. You have to go through the experiences of what learns and what works and what doesn't. And so as we've gone through these multiple programs, as we push them towards the clinic, it's been really important for us to to have that expertise, and we think it gives us a leg up versus other folks who may try to get into the space later and not have that capability.
Great. So I see we're at the 25-minute mark, so I'd like to thank you guys again for the great discussion and the overview.
Great, thank you so much. Have a good rest of the conference.